So, hello everyone. If this mic is bad, just tell me. So, okay. So I'm here with Marco to present to you our work about dynamics of tripartite synapses. So, what are tripartite synapses? They're simple synapses, so connections, chemical connections between neurons, but with this astrocyte wrapped around the synapse. Okay. So the astrocyte was usually thought to play only a supportive role in the dynamics of the synapse, but recent results show that actually it really can influence the dynamics of the neurons. So, for example, synchronization dynamics. So it can really regulate neuronal dynamics. So there is this interesting paper here that shows that in hippocampus, when you disconnect networks of astrocytes, so astrocytes can connect between themselves. When you disconnect these astrocytic networks, the neuronal dynamics changes a lot. So there's lots of properties in the neuronal dynamics that change when you disconnect astrocytes. So they really do play an important role in the dynamics of synapses. This is an experimental work which we aim in the future to try to replicate, or to simulate, to model. We didn't do that here because it's complicated. So we started with a simpler model and simpler mechanisms that Marco is going to talk about.
Yeah. Good morning. I will talk about the role of the astrocytes in the synapses. Essentially, the astrocytes receive neurotransmitter from the synapses, have receptors that are activated by glutamate, for example, and also GABA and ATP. And this process is calcium dependent and IP3 dependent because this receptor activates some catabolic process that produce IP3. This IP3 affects some calcium pumps and increase the calcium level in the cytosol of the astrocyte. This calcium level increase also redund in the release of gliotransmitters. These gliotransmitters affect the postsynaptic cell and the presynaptic cell, regulating the synapses. And also the astrocytes are connected by gap junctions. So this calcium and this IP3 can diffuse through one astrocyte to another astrocyte, activating synapses of so far of this area.
So just as a reminder, I don't know if everyone knows. So astrocytes are electrically neutral, so they don't have membrane potential. So when you analyze the dynamics, we analyze the calcium concentration dynamics. So that's the point, okay? So our first attempt at modeling this synapse was in this paper. It has some interesting dynamics, so IP3, glutamate, and ATP, which Marco mentioned. But there were some problems with this paper. So, for example, some parameters were definitely not clear. So we tried to do what we could, tried a few parameters, but we couldn't match the results with the same parameters. So this beta here controls the influence of IP3 in the calcium dynamics. And the original result was supposed to be that increasing the role of IP3 generates more calcium waves. This is calcium concentration, generates more calcium waves in the astrocyte. But we weren't able to replicate this. Playing around with a few of the parameters, we got close, but we still didn't manage to find this second spike here. So the astrocytic model in the paper was not really that good. And so since we didn't replicate the results, we changed to another model that had a few more mechanisms. Do you want to talk more?
Yeah. There are lots of interaction in synapses in real world, but we will focus in a single neurotransmitter, that is glutamate. Glutamate can be released by the astrocyte, and this can impact through the postsynaptic bottom and generate depolarization as the glutamate released by the presynaptic bottom. Also, this glutamate can impact to the presynaptic bottom, increasing the probability of the release of the glutamate, and we will model both process. And of course, the process of the single synapses, the normal synapses. And well, also the calcium dynamic and the IP3 dynamic. This is our model.
Yeah, so this is the figure that Marco wanted to put here. For me, it's too complicated, so I made a simpler diagram for you to understand. So there are two neurons and the astrocyte here, okay? We send a current to the neuron, a post current, that has some rate of spiking. So this input current makes the neuron spike with a certain frequency. And when we simulate the results, so our second model, we compiled a lot of different models from different papers. So there is not one single paper we can cite here, but the neuronal model is this one, so the integrate and fire model, adaptative exponential, so dynamics like this, which we already studied in LASCON. And the astrocyte model, so the astrocyte, follows this Lead-Intel model. The main equations are this one, so this is the calcium concentration, and these are the currents going into the cytoplasm. So these are calcium concentration that goes from the endoplasmatic reticulum to the cytoplasm. This is leakage from the endoplasmatic reticulum to the cytoplasm. And this is a current that goes from the extracellular space to the cytoplasm. And all of these currents follow a Hodgkin-Huxley formalism, which you can kind of see here. And they affect the IP3 dynamics. So IP3 dynamics also, there's the two influences. So IP3 also influences this current, which influences the calcium concentration. And the important thing to notice here is that the coupling between both neurons and the astrocyte is done through this term. So coupling is done in the IP3 dynamics, which goes then into the calcium dynamics. And so we found this paper which implemented some of the mechanisms we implemented already in Brian, so we used this simulator. Our first result implemented in Brian is this one. So this is the presynaptic neuron potential, postsynaptic neuron potential. And you can see here the square waves that we put in the presynaptic neuron. You can't really see this because this is wrong, actually. This should be seconds. So this is 50 seconds. So you can notice that the astrocyte is really slow. So it only spikes, so it has waves that are really slow. Slow to compare to at least neuronal dynamics. So this is not really good looking because the spike is so short that it doesn't appear in the figure. But we can see that with input rate of 1 Hertz, it generates an input and fire frequency of the presynaptic neuron of 1 Hertz. And this can generate calcium waves in the astrocyte. When you increase the firing frequency, so of the neuron, you can still generate calcium waves in the astrocyte. But if you increase further, you still increase a little more, and there is no more calcium waves in the astrocyte. So our first result here shows that only sufficiently slowly firing neurons can generate astrocytic calcium waves. Okay. We also actually tried to analyze synchronization between the two neurons, but at least for this adaptative exponential model, the firing rate of the neuron depends a lot on the input current. So when I don't know if you can see it here, but when the astrocyte releases, when there is a peak of the calcium concentration, it releases glutamate, and this glutamate changes a lot the firing rate of the second neuron. So what the astrocyte does in this case is desynchronize further the network. Okay, network and neurons, right? So Marco did a few more things with two synapses.
Yeah, in the case of two synapses, we have modeled two tripartite synapses connected by the gap junction between the two astrocytes. So as we see in this picture, we see that, well, we put a spike generating neuron 0, and this send glutamate to the astrocyte, activating and make the astrocyte do an oscillatory behavior. And the astrocyte also connected with another astrocyte, impact in the dynamic of the another astrocyte, also generate oscillation in astrocyte 1 in the another site associated with another synapses. And it's interesting because they have a time delay that is traveling for the wave from the astrocyte 0 to the astrocyte 1. And also this another oscillation generate gliotransmitter release and impact in the another synapses without a single common synapses between these neurons. So in this way, this tripartite synapses can affect another tripartite synapse through the gap junction between the astrocytes, generating this pattern. And we also simulate the behavior of 4 synapses. In this synapses, some complicated graph, but we can say that in this case, we have connected all the astrocytes with several connections, gap junctions, and also neuron 0, more complicated the topology. But we realized that the astrocytes are synchronized in some aspect. Look, the calcium wave is like similar in the four astrocytes. So we have some synchronization of our astrocyte that reduce in that this astrocyte release gliotransmitter at the same time. Well, not same time, but it's like more stress the delay between these gliotransmitter release, then also impact in the neurons generating this bursting behavior. It's like a synchronization with the astrocyte, then generate also an impact in the dynamic of the neurons at the same time or close time at least. Then I proceed to cut these synapses, these gap junctions, and inspect what happened with the synchronization of the astrocyte. And when I cut some synapses, some gap junction, the synchronization is also happening. But in this important connection, the synchronization is affected negatively. So we have a time delay also in the gliotransmitter release that also impact in the neuron dynamic, and that's happened when you put some important connection in this topology.
Yeah, so to sum up, for a single tripartite synapse, we showed that slowly firing neurons can affect the astrocyte, generating calcium waves. But if the firing frequency of the neuron is sufficiently high, that doesn't happen, and so the astrocyte is not really affected by the neuron. And another thing we saw is that disconnecting astrocytes, at least in a certain way, can change their dynamical behavior. Okay, and of course, when the astrocytes lose synchronization or their dynamics change, the neuronal dynamics will also change because that is controlled by the glutamate release. So this connection of astrocytes, like in that previous experimental paper in the hippocampus, can change the dynamical behavior of the network. Okay. And so, as a final remark, we like a take-home message for people who have slept through this presentation. We just like to say that astrocytes can be really important for neuronal dynamics and neural networks. So be careful with that. And so for the future, we suggest to Roque that the next event be called LASCON VIII. Okay. So thank you very much.
We have one question. Yes. Hello. We have one question from Leonardo Dalla Porta Dornelles. His question is why when calcium peak concentration release glutamate, the neurons get more desynchronized? Is it because the neurons fires in bursts?
No, they do fire in bursts, but that is not necessary. It's just that in this model, the adaptative exponential integrated fire, the firing frequency of the neuron is highly dependent on this input current here, highly dependent. So when the astrocyte releases glutamate, of course, it generates input current to the neuron, and this changes the firing frequency of the neuron. But when I say neuron, I mean the postsynaptic. So astrocyte waves generate input current to the postsynaptic neuron, but don't affect the presynaptic neuron. So it changes the firing frequency of just one of them. So this synchronizes the two of them. Okay.
So you used an exponential integrated fire type model for the neurons and the conductance-based model for the astrocytes. Do you think that mixing this heterogeneous type of model can have some impact on your dynamics? Why you chose exponential integrated fire rather than, I don't know, Van Budzinski or Oshkin-Axley for the neurons?
So I showed here a spike, but the interesting thing about this adaptive exponential is that it can burst. Actually, the pictures are not really good, but in here there's bursts when the input current lasts for sufficiently long, right? So I forgot to say, but in the hippocampus experiment, the neurons were bursting. So we're thinking like in the future, we want there to be burst dynamics. So that's why I chose it. I mean, I don't know how it will influence with another model.
Yeah, you said that astrocytes have a neutral membrane potential. That's wrong. I mean, every cell has a membrane potential. It's just that membrane potential doesn't play any role. Can you come back to the equation of the calcium dynamics? Is that the way people actually, model these situations? Because my feeling is that calcium takes time to diffuse. I mean, here you're assuming that everything is point-like. Either IP3 that diffuses fairly fast with respect to calcium is the signal that goes from one astrocyte to the next, and in that case you can go fairly fast. If it's actually calcium that has to diffuse, you should have strong diffusion effects, I think.
Yeah, in our case, the equation for the diffusion between astrocyte to astrocyte, we consider the IP3 diffusion because we know that calcium diffusion is more slow. Well, there are some hypotheses that probably the calcium diffusion as the slow could be. I read that. It's like, well, the calcium diffusion is slow and gets some, but as it's slow, these have accumulated behaviors from another astrocyte, but
Okay.
it's like, well, it's an hypothesis already, and we work in our equation with IP3 diffusion.
Okay. So, but where is IP3 diffusion in the equation?
The IP3 equations, well, we don't have in our slides now. Because we have a lot. This is only 6 equations. We work with 10 more equations,
okay.
and we isn't on our slide.
Okay, I think it's better to finish now. It's any more questions? No one no. So okay, let's move to the next presentation.
Five minutes.
You can start. Let's wait until 9:20, so we will respect people who are.
Just a second. So, hi, I'm Natália Matos, a master's student in the Brain Institute back in Natal. And this is Roberto Budzinski, a PhD student at the Federal University of Paraná. And today we are going to talk to you today about our project entitled Modeling Gamma Frequency Adaptation in Neural Systems. But before I start, I want to thank Antonio Roque for organizing this summer school. It has been a fantastic experience so far bringing so many people together, so thank you. Well, so proceeding with the project. Back in Natal, I study with Sérgio Neuschwander, and we study mainly the electrophysiological activity from awake and anesthetized cats, as well as awake captain monkeys. Here we can see one of our devices. So this is a head fixation apparatus. This is a recording device. And we use quartz electrodes to record the activity from different visual areas, including the retina, the geniculate nucleus in the thalamus, as well as the visual cortex. And our visual stimuli that we apply varies from a wide range, from ratings to more complex stimulus like videos of natural scenes. Our trials take around 5 seconds, and the animals have to fix the gaze during the experiments. This is an example of one of our results. This was recorded from the retina of anesthetized cats. And the stimulus was just like a big circle, very luminous circle. And although there are many interesting features shown here, I want to highlight two. The first is the strong gamma oscillation. And the second one is the non-stationarity nature of its frequency. So it starts at a higher value, the activity of the frequency, but then it quickly decays to a lower value. And yeah, in gamma rhythm, I don't know if you remember from classes, but it's a pattern of brain activity, of oscillatory activity that goes from between 30 to 120 hertz. And this frequency is not constant here. In this particular example, it starts around 90 hertz, and then by the end of it, it decays to 70 hertz. And interestingly, this phenomenon is not restricted to the retina of anesthetized cats. It can also be observed in the visual cortex of awake monkeys. In this case, the decay is less steep and the frequencies are lower, but we can still see it happening. So in the start, we have around 60 hertz, and then it decays to 60 hertz by the end. So it takes some time before the frequency stabilizes. Well, but then we can ask, why is this interesting? Why do we care? And when we look at different cells in the visual pathway and the latencies of response, there is the time it takes from a given cell in the visual pathway to fire its first volley of spikes. We can see that these latencies are very short. Electrophysiological studies show us that a neuron in the inferior-temporal cortex is able to fire selectively to faces within 80 to 100 milliseconds after stimuli presentation. So the visual information has to travel through many layers of the visual system and very quickly. But as I previously showed you, it takes hundreds of milliseconds before the frequency stabilizes. So this might have some impacts. Oh, and this is happening in widely different circuits. We are seeing this in the retina of anesthetized cats, as well as the visual cortex of monkeys, where the mechanism behind the generation of gamma oscillations are believed to be very different. So, yeah, so what is happening? Like this might have some implications for theories of the function of gamma oscillation. So in the context of visual processing, gamma is believed to work by binding different features of visual stimuli, as well as communication or information flow through the many layers of the visual system. And then this might have an impact on the series. However, this phenomenon has not been well studied yet. And that's why we chose for a project to try to model this phenomena by doing a model that simulates gamma first in the frequencies close to the ones that I see in my experimental results, and then try to simulate this decay and see some of the possible mechanisms behind it.
Okay, so to start, we take a model to simulate the neural behavior, and actually we used integrating fire model. I think it's the same that Kalel used it. And here we have a conductance-based exponential integrating fire model. And the first equation here is related to the membrane potential of the neuron. And the second equation is related to an adaptative in the spike rate. So here in this graph, in this graph, we can observe the dynamical behavior of the neuron using this model. So as a first approach, we use this model and try to build a network to generate gamma oscillation to understand the mechanism behind the frequency decay. However, we are not able to produce stable gamma oscillations and even to study the decay on the frequency. So, can you as a second approach, we tried a simpler model, and we avoid the equation of the spike adaptation and just considering the equation related to the membrane potential. So here we have the dynamical behavior of this kind of model. We can see a set of spikes, and in this case, we can control the rating, the rate of the spikes, changing the parameters. As Kalel said too, this model is very, we can change the frequency that the neurons, the activity of the neurons, changing the external inputs in the model. We can change the rate in the spike activity, changing the conductance parameters and the capacitance, for example. So now that we have a model that depicts a behavior that we would like to understand, we can try to model a network to generate gamma oscillation, and then to try to understand which kind of mechanism can produce the decay that we observed in the experimental results. So I think several lectures showed this kind of model, and the first approach to build a network, we use an excitatory and inhibitory population to build the network. So here we have an example, what is the network. So in our simulation, we're considering 100 neurons, where 80 are excitatory and 20 are inhibitory. And here we have an external input in the excitatory population, and we have connections between the excitatory neurons. We have connections from the excitatory neurons to the inhibitory neurons. We have connection between the inhibitory neurons, and we have connections from the inhibitory population to the excitatory population. And here, of course, we have a set of equations to each neuron in our network. And here we have, besides the external stimulus, we have the synaptic current. In this case, the synaptic current can be excitatory or inhibitory, okay? And also, we are considering a kind of white noise in our network in order to have more realistic results. And here we have, to do the simulations, we used NEST simulator. So here our synapses are alpha synapses. And in this case, we can control the time delay of the synapses and the synaptic strength. And here we used random topology to build in the network. And so we have the random matrix to the excitatory case and to the inhibitory case. And we use the probability of connection equals 0.5 in order to simplify the model and try to understand the mechanism behind the oscillations. So we set the parameters to that the inhibitory neurons depicts fast oscillations around 80 hertz. And the excitatory neurons depicts a rate of 40 hertz. Of course, these numbers are related to the isolated neurons. When we consider the entire network, we can see here, this is the spectrogram of the spike activity of the network, and we can see that we can produce gamma oscillations around 50 hertz, and these oscillations are stable. So here we have a raster plot of the network. The blue dots represent the excitatory neurons, and the orange dots represent the inhibitory neurons, and we can observe that the frequency is stable. So now we can try to understand the decay on frequency that we observe in the experimental results, and we change the synaptic strength in the network. In this case, we increase the synaptic strength of the connections from the inhibitory populations to the excitatory populations. So we are increasing the inhibition over the excitatory population. Here we use exponential function. So here we have a representation of the synaptic strength increasing as a function of time. And here we have the experimental results, the mean frequency of the experimental results, and we can observe that the decrease from 50 hertz to around 40 hertz. And here we have the spectrogram of the simulated results. So we can see a good agreement between the simulated case and the experimental results. We also analyze the synchronization during this process. So in order to measure the synchronization, we wrote a code to analyze the synchronization error in the network. And to do that, we consider the membrane potential of each neuron. And here we are evaluating the standard deviation of the membrane potential, considering the mean value of the mean field of the network. So here, V, overline V, represents the mean field of the network. In this case, S represents the standard deviation. And if we have a lower value of the S, I mean, if S depicts a value close to 0, we have almost completely synchronized network. And here we consider the relative error because we divide the standard deviation by its mean value. And here we can observe during the process of the frequency decay, that the error in the synchronization also decrease. So we can observe that the network depicts a higher synchronized state at the end of the simulation. So when the frequency is lower, the network depicts a higher synchronized state.
As I have said before, this phenomenon is not restricted to the cortex, where ping models are believed to be one of the main mechanisms behind the generation of gamma. So that's why we also wanted to try in a different model. So in this case, we are doing an inhibitory network. So we have two populations of inhibitory neurons. They are connected between themselves and with themselves, and we have a strong drive coming from those two populations. And yeah, so in this case, we are also able to observe gamma oscillation around the frequency that we see in the retina back in our experiments. And here are the two populations, the Raster plot of one population and the second population of inhibitory neurons. And then we try to also simulate the frequency decay. This is our experimental results, a mean of our experimental results. And in this case, to simulate the decay in the frequency, we changed the synaptic weight to increase the synaptic weight between all the connections of the network. And by doing so, we are also able to get some results that fit with our experimental data. But when we look at the synchronization differently from the previous result, we could not see a change here. Yeah, it's kind of stable all around the simulation. So yeah, so these are our results. We are able to simulate gamma in a ping and an ing model. We were able also to simulate the frequency decay by changing the synaptic weights. And for the future, it would be interesting to try this on more realistic models, for example, of the retina, or maybe from V1. There are many models available. We just didn't have enough time to explore them here. And look further for mechanisms like short-term plasticity or another thing like that that could be causing this decay in the frequency more realistically. So that's it. Thank you.
Okay, you also finished before the 50 minutes, 10 minutes, 10 seconds before. Let's go for questions. 5 minutes for questions. No questions. Okay.
So you kind of considered, but then kind of rejected neuronal adaptation as a mechanism from looking at the single neuron, the exponential integrating fire. But when you have the circuit, the ping model, that probably is what's at play, right? That you put in a stimulus, the activity goes high, and then you kind of start introducing these adaptation currents, spike frequency adaptation in the neurons. And so, I mean, do you think that it would be worth kind of reintroducing in the spiking circuit model, the ping model? Or do you have a reason to think that this strengthening of the I to E synapse is actually like a plausible kind of mechanism of what's happening during this adaptation?
Yeah, so we have tried to model the firing adaptation before, but we couldn't have results. Like we could simulate the frequency decay. That's why we chose not to use it by now. But yeah, it's still a valid mechanism, possible mechanism. Maybe we just need to work with the network a little bit further, a little bit more. And from now, we don't know like what is possible and what like, this is still a possibility. We don't know if we could discard this or no, this is not happening in the visual cortex, this is not happening in the retina. It's possible. And the synaptic strength, I think that maybe it could be a process of short-term plasticity, like I mentioned. Maybe this is happening there, so that's why we try to change the synaptic weights first. But yeah, if we keep going this work, we're going to try different ways as well.
The change in weight that you use is fairly large. I mean, you have a factor of 5, at least 5 between the beginning and the end. Is it something you tried just at the beginning, or you tried several possibilities to get adaptation of frequency?
Yeah, we tried several values. Actually, I didn't show, but the synaptic strength between the excitatory population, for example, I mean, the inhibitory population started from 30 and go into 120. And the excitatory strength is like 20. So it's not that high, it's like 6 times in the end of the simulation. But I mean, we tried different values, and this is the best fit that we can obtain using this model. Of course, it's just a first step, so to try to understand the mechanism. But we did try different values, and this is the best fit that we had.
So if you think of the physiology of the circuits, the type of circuitry you have in the cortex, where you may record the adaptation, is pretty different from the one you have in the retina. So have you thought about how you could check different hypotheses and adapt your model to the different situations?
Well, yeah, this was the first idea. So we wanted to try to model more realistic models because we know that the retina is generating gamma in a totally different rate than the cortex, right? In the cortex, we have interneurons, pyramidal cells. So, yeah.
How would you modulate? In retina, you have electric synapses that
Yeah.
mediate the interneuronal, the
And
lateral interneuronal. So how could you adapt an electric synapse as you do in the retina?
Yeah, and we also see in the experiment results that the frequencies are much different, the decays are much different. So yeah, there's probably something. The decay still happens in both systems, so that's interesting. But yeah, right now we don't have a specific hypothesis for the circuits. The cortex maybe it's a little bit more straightforward in a way that these ping models make more sense there, but then for the retina, there's still a lot that could be happening. And for now, we don't have any good hypothesis why this is happening in the retina specifically.
Okay, so good morning. I'm Leticia and this is Eric. And today we're going to present the results of our project developed here in LASCON, analysis of synchronization parameters in an oscillatory network using NEST. So the normal brain function depends on the integrity of the dynamic connections between neurons and regions. And those connections depends on the structure that comprehends the anatomical structures that make these connections possible, and also the function that is the process of transmitting information. And the synchronization can be inside a small network between close neurons, and also a long-range synchronization. That is a coordinated communication between distinct regions. These alterations in connectivity can lead to pathologies, and a lot of neurological pathologies have this kind of alterations, including the epilepsies. So in the epilepsies, there is abnormal synchronity patterns that depends on the time that the EEG recordings are being made. If it's made during the epileptic seizure, on the ictal period, there is an increase in the synchronization, but in the peri-ictal period, around this epileptic seizure, there is a decrease in the synchronization, which made studies to believe that a seizure is just the tip of the iceberg, and that this smaller synchronization could be a mechanism of anticipation of the seizures, therefore making it a mechanism of like anticipating the seizure and being something that could be changed to prevent a seizure to happen. In this case, deep brain stimulation has been studied as a mechanism of prevent the initiation of the seizures. So, as targeted stimulation through deep brain stimulation, for example, has been studied for modulation of brain states and a treatment for a group of neurological diseases, including epilepsy and others. Although many aspects of the impact of these focal stimulations in a network level remain unclear, it must be studied. So in this project, we aim to create an oscillatory network in order to analyze the normal synchronization patterns of the network, and also evaluate the effects of disturbances in specific nodes in these networks, considering the disruption of connections between nodes, and also the input of an external input that is either negative or positive.
Our hypothesis is the synchrony between different brain regions is a measure of the global effects of a focal stimulus. So these disruptions of connections between crucial nodes of the network can change in synchrony. So for the construction of our network, we use the McCake macro connectivity database, COCOMAC. So we have the adjacent matrix. With the adjacent matrix, we have the areas of the macaque brain. So we have the connections of these areas. We use this adjacent matrix with the nest for create the network. So with the structure, we can create the network. I will talk more about the structure network later. So for dynamics network, we chose the nonlinear rate model with sigmoid external inputs. We chose this model because there is a reduced number of equations. So we have a lot of areas. We have 242 areas. So we need a model more simplified for construct the network. So let's talk about the structure of the network. We have 242 brain regions of the COCOMAC. The edge we construct based on adjacent matrix. I will talk more about the adjacent matrix. In the network dynamics, we use the computational model, nonlinear rate model. Let's talk about the connection. With the nodes are connected, we have this model. So each node have a excitatory population and the inhibitory population. These populations are connected with each other, so we have a excitatory population connected and analyze the connection with the nodes. We have the excitatory population with the node 1 connected with the inhibitory population in the other nodes. So we have 4 connections, 2 in the excitatory population, to the excitatory and inhibitory, and 2 connections for inhibitory and the other populations.
So as we have 242 nodes, we had to filter a little bit which nodes we would explore. And so as each node represents a different structure on the macaque brain, we chose the hippocampus node, that is node number 58. This node has 18 edges, and one of those is, and hippocampus is also connected to the temporal area, that is node number 18. Those regions were chosen because they are very important to the pathology of temporal lobe epilepsy, and mainly the hippocampus, which is very disrupted during the physiopathology of epilepsy. And for the measure of synchronization between these two nodes, we chose the phase locking value, which measures the phase synchronization between two oscillatory signals, and is often used as a representation of the functional connectivity between two nodes. So this is a normalized measure that takes values between 0 and 1, where 0 represents no phase synchrony between the two signals, and 1 represents that the relative phase between the two signals are identical. So it's like the perfect synchronization between two regions. And those results will be shown in 2 ways. First, a bar plot of the phase locking value for each of the conditions we will show later, and also some histograms of the difference of phase between the two regions that will show if those nodes are synchronized, in which phase they are synchronized.
There is our network 1. In the left, we have the adjacent matrix. So each dot, yellow, corresponds to one connection. So we have the 242 areas, 242 areas. Here we have if this area are connected with this area. So we use the adjacent matrix for building the network. This network, there is many of nodes. Each node, there is a number of edges. So we have this node. These nodes have a lot of edges. These nodes are less connections.
Okay, so first for this normal condition, I bring here the few plots. And as we can see, there are a high quantity of peaks, which means that those two nodes, the hippocampus and the temporal area, do not have, in this normal condition, a very high synchronization between them.
Question 1. What's happened if we disruption connection of the hippocampus? So we have our network, and now we go cut these connections. So just represented we have the same adjacent matrix, but now the node corresponded hippocampus. All the run and column are equal to 0.
So for this condition, the same plots for the excitatory and the inhibitory populations, we can see there is a higher peak at the excitatory population, but there are still a high number of peaks, which means that those structures are also not very synchronized.
Second question, what's happened if we input a external current positive in the node corresponding in hippocampus?
And so for this condition, we see a smaller number of peaks, but the proportion of the trials that correspond to each phase are also not high. So this probably means that this synchrony is also not very high. But with the PLV plot we will show after, this will be more clear.
The last question, what's happened if we input an external current negative in the node corresponding at hippocampus? What's happening with synchronization?
So the plots are very similar to what we show with the positive input. As we see the smaller number of peaks, but also very numerous, so probably the synchronization is not very high as well.
So let's analyze the 4 situations with the phase locking value is a absolute value. So we have the normal network, the network with without the hippocampus, the network with the positive inputs, and the network with negative inputs. We can observe it, the phase locking value decrease in the synchronization, the disruption of the node corresponding hippocampus, but there is a higher value in the positive inputs and negative inputs. So there is difference in the synchronizations with these 4 networks.
So to conclude our presentation, we were able to show through our nonlinear heat model that it's possible to analyze the oscillation patterns of this network and the synchrony. But, and the model that we used was effective to this study, but we would need more robust information about the connections between these 2 nodes to replicate a more physiological pattern of connectivity between these 2 areas. So these results could be more trustworthy.
It's important to say there have a lot of parameters with searching literature, so there is more studies about these parameters.
So we want to thank everyone for paying attention to us, especially Roque for developing LASCON and accepting us here, and also the tutors that helped with our project and made it possible. Thank you.
Again, you finished in time, 44 seconds before, so we can proceed to questions.
How do you justify the long-range inhibitory connections?
Sorry?
How do you justify the long-range inhibitory connections? I mean, the inhibitory connection between nodes.
You mean?
So when you explain how you couple the nodes, you explain that the excitatory population of one node synapses onto the excitatory population of the other node and on the inhibitory one. And you also explain that the inhibitory population of one node send axons to the other node. I mean, how do you justify that? I mean, as far as I know, there's only long-range excitatory connections, no?
Yeah, this was something we were talking about because I I thought it would be, the oscillatory connections would be from longer range, but the inhibitory connections are used restricted to the area. So I don't know. We were talking about cutting this because this didn't, I thought it would be better, but in the end we didn't do it. So sorry.
There is a long range inhibition. It's a very specific system. They don't have everywhere, but in some hippocampus notably have quite a lot of long range inhibition. It's a new thing, but it's not global. Long range means even this 10 centimeters, 15 centimeters. It's a new thing. So all your weights were identical?
No.
No, okay.
The oscillatory, the inhibitory weight was 5 times
No,
stronger.
I mean the long range weights.
Yeah.
Did you use a binary connectivity matrix? Okay.
Yeah.
So and
inside the area and between nodes, it was the same.
And do you, and for the mass, for the neural mass, you fix the parameter once for all, and then what was the dynamic of an isolated node? Was it oscillating? Was it?
Yeah.
Okay.
Yeah, there was. We plotted the rates, but we didn't show here. But yeah, it was isolated.
And what is your interpretation of your result? The stimulation increase synchronization. Is this a good thing or a bad thing?
Yeah, it depends. For example, considering epilepsy, if there is an increase in synchronization during the seizure, it's bad. But considering that the periictal periods have a decrease in synchronization, it would be, and it could be a predictive value for the beginning of a seizure. It could be, it could have a biological and also medical value to prevent a seizure to happen. If this is a pattern for all the seizures of a patient, for example. So if an external input could restore the network synchronization, for example, it could prevent a seizure to happen.
So you focus a lot on hippocampus as a site of stimulation. Presumably in the large-scale network, it is maybe more connected or it has different topological properties compared to more peripheral nodes like primary visual cortex. Have you thought about, or do you have predictions for, whether different sites of stimulation would have different effects of the synchronization, like a more peripheral node versus a more hub node?
In the first, we think analyzing more nodes, more situations, but we have no time to explain this very situation, so we focus in epilepsy. So we focus in nodes corresponding to hippocampus, but we saw the other nodes in the other situations. The model is possible to do this.
We thought about exploring other areas and other node connected to hippocampus and other structures that are also represented on this matrix, but we wouldn't have the time to develop, and we had some technical issues, so that's what we could do.
no. No, le rápido. So, can we start? So we are Fer and Danny, and this is the title of our project, is Bifurcation Analysis of Alternated Rhythmic Oscillation in a two-cell network of mutually inhibited non-oscillatory neurons. So to start, I want to define what is a central pattern generator, which is a neuronal circuit that, when activated, can produce rhythmic motor patterns such as breathing, walking, and these patterns generate in the absence of sensory or descending inputs that carry a specific timing information. So there are two main mechanisms that are proposed to the generate of these rhythms. One is in the presence of a pacemaker, that is a neuron with the intrinsic properties of oscillate, that when it's coupled with another neuron that has not this property, they drive the second neuron to oscillate in an antagonist way. And the other main mechanisms is not throughout the intrinsic properties of the neurons, but about the coupling between neurons. And this case is in the reciprocal inhibition of two non-oscillatory neurons. And the biological basis, for these mechanisms could be that one neuron that, when it's excited, inhibits the second neuron that, when it's inhibition, has a postinhibitory excitation that inhibits the first neuron, and so on. So this is the model that we are going to use in the project. These are two-cell network with mutual inhibitory connections between them. So to do this, we base our work in a recent paper where they demonstrate that throughout synaptic connectivity, they can, in non-oscillatory neurons, they can produce network oscillations.
Well, here, we have here a Hodgkin-Huxley type equation where Rodrigo and Kalel explained this well. Now, in this equation, we have a current, lead current, potassium current, and synchronization or synaptic current. This model is like a potassium conductance-based model. But, so, to develop a model inspired or based in this work, where but now we want study and explore the calcium current. This is the Hodgkin-Huxley type. We have a lead current, calcium current, apply current externally, and current in synapses. synapses coupling current. We can see in this model very similar the Hodgkin-Jackson type. This is the parameters of gating variables. Sorry. Well.
So to develop the method, we use we integrate with Ronchakuta 2 degrees, and for that, we implement the model in Python, and then to perform the bifurcation diagrams, we use the PDS tool. So important in this model, for each neuron, the equation takes in account the effect of the second neuron. Throughout this parameter, that is the coupling within the two neurons. So in base of this, we found 3 different scenarios in which we explore the dynamics of the network. So the first one that we obtain is this dynamic where when the neurons are not connected, they're not oscillating, and when they are connected, start to oscillate in the antiphase, and this can be explored in the phase space, in which it's possible to find a limit cycle. So these are the parameters that we use. And we did the same thing in the PDS tool to reproduce the model. And again, we can see that when you connect the neurons, they start to oscillate in antiphase. The difference with the Python model is that in PDS tool, you cannot put a lag between the connections, so the neurons are connected since the beginning, and you only analyze that moment. And this is the bifurcation diagram where there is a half point, and there's a unstable and a stable branch. And for this second one,
Great. In this second behavior, we can see the behavior when put current apply external in this value. We have 2 different neurons. When coupled, the amplitude is bigger and make antiphase. See? Yes. Well, when there's a couple, back to the same behavior and persistent oscillation. We change the current, the calcium current and the applied current in this case. In the next one. In the PIS tool, we replicate this parameter and make the same behavior, and we obtain this bifurcation diagram. We can see an unstable line and little point of Hopf bifurcation, limit point, and branch point in this case.
And the first one is when you have an attenuation oscillations. That is when you connect the neuron, when you apply the current to the neurons, they start to oscillate, but after that, they don't oscillate anymore. And you can see this in the phase space that they try to converge to a fixed point. And again, this was assessed in a PDS tool. And in this case, we only find bifurcation, half point. So the conclusions is that, it was validated that this two-set network model can be used as a pattern generator mechanisms, and the parameters that can affect the dynamic of the network are the
current applied, the conductance of calcium, and also the coupling between the strength of the coupling between the neurons. And also it was confirmed that the existence of limit cycle and half bifurcations. So we want to thank Mauricio, Horacio, Vinicius, and Milton. And Rodrigo, who is a postdoc of Horacio. So that's all.
Well before schedule, sorry. And so we have lots of time for questions. Questions? So you replicated a previous model using Python and PyDS2. What have you learned in this replication work that you have done?
I mean, basically, I haven't programmed before in Python, for example, and I learned how to program in Python and also to use the PDS tool. So I think that it's a great experience, at least for me.
Well, PyDS tool is a very hard tool and have a lot of things. But when you develop in Python, you need to make a lot of code. In PyDS tool, you need a little bit of code, and you have a function very fast and very complete.
I meant in terms of the model, in terms of the scientific content of the model, not in terms of the tools that you used. If you had to say improve the model, what would you do?
I mean, we need to find the null clients and also the maybe to perform a limit cycle.
This also would be in terms of analysis, but also, I mean, in terms of improving what the model is good for.
I think that the model is working properly. I mean, we could reproduce 3 different scenarios, but I think that we need to...
So rephrasing, how would you extend the model then, if you want to add other elements?
Yeah.
So this two neuron model.
Yeah, maybe we can add another neurons or, I mean, yeah.
Another channels.
Yeah. Not only the channel, the calcium channel, maybe.
Any questions?
So can you comment a bit on the bifurcation diagram for the second or the third regime, the one where the one where you didn't have any stable? This one probably. Can you comment on what is the meaning of all these various points? Because you went very fast, so I'm not completely sure to have understood it.
The P1 and P2 are the points of the equilibrium point. So you construct that curve, and then after that you try to detect the changes of the equilibrium. So first you can detect, for example, a branch point. There is they give you the angle of the bifurcation. And you can also detect, for example, the presence of limit points, which are delimited the limit cycle. And also you can detect different types of bifurcation. In this case, you can set all, for example, and they find you all the bifurcation that can be present. In this case, it only detects half type bifurcations. So, which was what we expect to find in base of Horacio experience.
Of course, the 3 different cases give you the different landscapes of critical points. So could you understand in some way how you pass from one to the other? Or did you study just these isolated examples?
Yeah,
because of course here they are different, but you could have maybe some kind of interpolation between them. Yeah. So do you understand the logic of one diagram transforming into the other?
No, we didn't do that. We don't have too much time, and the PDS tool consume a lot of time because you don't have a really good tutorial. So, I mean, we explored these parameters that I think that is interesting. I mean, it's like an opposite condition. In the first one, you don't have any oscillations when they are not connected, and the other, they are oscillating, and they start to oscillate in antiphase. So for us, it was interesting to start these three conditions. But yes, it can be done. I mean, there is a lot of work to do. It's just the beginning, I think so.
Okay. Maybe just the last question. So have you thought about, so of course the dynamic behavior that result from these different bifurcation diagrams are different. So you started with the scientific question of studying a CPG and CPG dynamics. So have you thought about what could be possible functions of these different regimes that you consider, for instance? So could you exploit for different functions the different properties of these points? Have you?
Yeah, I think that, in this case we only used as a parameter the strength of the coupling, but I think that you can use other parameters. I mean, maybe the.
I mean biological in the sense. Now, okay, that's mathematical model, but it's
Yeah.
mathematical model of a neural circuit. So the neural circuit does different things. What could be a function, for instance, of using CPG in this regime with a branching point with respect to using having a CPG like the previous one, which didn't? So what could be a usefulness in biological sense of these two different cases?
I think that the first use that that I understand that they have the model is to understand what is the function of the calcium channel. After that, we try to play with another parameters, but I think that mainly is to explore the calcium function in this network.
I'm waiting for Horacio's question. He wants to make a question, but he didn't send it. There's only 4 minutes.
Right.
To answer Damien's question, in the stomatogastric ganglion of a lobster, so the system that was studied by Marder and other people, it's known that the muscles regenerate different rhythms at different stages of the digestion, and that's why you need to have different rhythms generated by the CPG in that case.
Synapse. Horacio.
Hi, from Horacio. There are 3 regimes for the single oscillations: stable nodes, stable foci, and limit cycles. The 3 of them produce network oscillations. Have you seen any difference in the bifurcation diagrams between the 3 different cases? Apparently it was too long. Yes. That was his question. That if you saw any difference between the 3 different cases.
Different diagram.
Oh. Okay, good.
Hi? No. Oops, but for some reason the connection. He said the question is
Yes,
posted.
yes. Oh, it's what I.
Yeah, Newton has already. I think he's watching us, no?
Yes, he's watching.
Yeah. So can you answer the question?
Did you understand?
What is the question? Sorry.
difference
Speak in the microphone, please.
What is the difference?
Between the three different cases.
Because we changed the parameters and we changed the stability, and the diagram bifurcation changed. It's different stability in different case.
Yeah, okay. I mean, he said he couldn't post it. I couldn't let you know, but I mean, I think he posted it, no? And you responded. So I suggest that you talk to him later and then you discuss this issue, because now we don't have time.
Right.
We have to proceed to the next presentation.
Oh.
Okay, anyway, thank you.
Yeah.
Tem que falar com ele, porque esse tipo de...
Usually, if
Okay, you can start.
Okay.
in a full.
How to play this? Sorry. Thank you. Hello, everyone. I'm very glad to present to you our work about exploring the effect of stroboscopic light to the output of retinal direction selective circuits. The stroboscopic light is quite a long name, so I'm going to call it a flash. Basically, it's a regular flash. Our work is inspired by some discovery made by NASA. So they sent astronauts into the space, and the astronauts have motion sickness. So they want a solution, and surprisingly, they found a single ambient flash of 8 Hertz can cure the motion sickness. So they give you a flash, and boom, you don't have motion sickness. And how does that happen? We don't know. What we know is motion sickness is caused by conflict from input of a visual system and vestibular system. Basically, if what we see doesn't match what we feel in the inner ear, we have a conflict, then our brain is confused, then we have motion sickness. And the only way that the flash can affect us is through the visual system, which is our retina, our only light-sensitive organ. And in the retina, the motion is detected by this direction-selective retina direction-selective circuit. And this circuit involves 4 cell types: photoreceptors, bipolar cells, SAC as stable summacron cells. I'm going to call it stable cells, and DSGC as direction-selective ganglion cells. I'm going to call it DS cells. So it's a complex network, they connected in a complex way. Photoreceptors provide glutamatergic input to bipolar cells. Bipolar cells can provide glutamatergic input to stable cells and DS cells through AMPA and MDA receptors. And stable cells, the most interesting guy here. It has 2 neurotransmitters, excitatory one, acetylcholine, and inhibitory one, GABA. And it is a non-spiking and axon-less neuron. So it doesn't have spikes and it doesn't have axon. Everything happens in its dendrite. So it receives input on the dendrite and also makes synapses to provide output also on the dendrite. And finally, the DS cell is a ganglion cell, which is the output neuron of the retina. The axon send the signal to the other part of the brain. And it is called direction selective because it has direction selectivity. In this way, it has a preferred direction and a non-direction. And if the object is moving in the preferred direction, it fires a lot. Where in the opposite direction, it fires very few. So our job is to build a model for the retinal direction selective circuit and to see how the flash change the output of this circuit. How are we going to do that?
because in retina, it's not only about who connects to whom, it's also about like where they're making the connection and how the connection is organized in space. And like what kind of, their morphology also matters to the computation. Therefore, we cannot just build any kind of model. We have to build a detailed model, including all the morphology, ion channels, distribution, or something. How are we going to do that? So we find out the channels existing on the dendrite of both cell types. You can see all kinds of sodium, potassium, calcium channels, and they are not evenly distributed, some on the soma, some on the dendrite, and all kinds of details. And we found the morphology of stable cells and DS cells. We put all the mechanisms, all the channels, neurotransmitters, receptors on the cell, and we couple them, and we put more cells to form a network, and boom. Guess what? Because there are like 90 stable Zamacron cells connect to a single DS cell, and that's what it looks like. It's a mess. So I think it's not a very wise choice to do this kind of project in 2 weeks. So instead, we're not doing this. We're doing our simplified detailed model of retinal direction selective circuit. And how we're doing this, we start by modeling the bipolar array. It's not nothing here. That's our bipolar cell. It might be a huge simplification, but we use this circle to represent bipolar cell. It has properties. Actually, it doesn't only represent bipolar cells, it represents a unit comprised of photoreceptors and bipolar cells, because these two as a unit, their response is quite stereotyped to light. Basically, in the on pathway, the bipolar cell release glutamate when there's light. And this cycle actually, the size of the circle actually represents the size of the axon field of bipolar cells. That's what it looks in real retina. It's an irregular shape, and it tiles the entire retina like a mosaic. And also how we make this thing responsive to light. We use a neural function, light stim, which is a point processor, and basically we translate the light stimulation pattern to a sequence of time, and we give the sequence of time to light stim for it to send a sequence of event to trigger the release from the synapses. And that's how, so now we have a bipolar cell. Oh, it's gone. It's still there. We have our bipolar cell, and then we tile the entire space with a random array using Python disk sampling and reaching a realistic cell density, which is about 5,000 cells per millimeter square. So now that's our bipolar cell array. And so let's see, we'll talk about how we simulate stable cells.
Yeah, basically here how we did the simulation of the stable cells. First, we looked for a biological morphology of the cell. As we can see in the morphology photo, it's a cell that on a plane it has a radial symmetry, and the dendrites have a lot of branching points as far as they are from the soma. And that's what's the actual model that we used, that we found on data sets. We added the calcium mechanisms to the cell. We have two calcium mechanisms and the leak mechanism. First, the KV3 mechanism, it's a voltage gauge channel. It's basically, it activates during the polarization and is responsible for this response on the cell that saturates when it gets excited, and when it gets excited. And the KIR also potassium channels, and the function of these channels basically are this, they saturate the hyperpolarization. Here we have experimental data of a real cell and our simulation data. Here we added a current on a cell, and we recorded the response on a dendrite. And as we can see, we were able to fit with a relative success our model. So the next step is to connect the bipolar cells and the stable cells. On these papers by Russell, we found the relationship between the connections and between bipolar cells and stable cells. Basically, this blue line shows that on the first 100 micrometers, we have the connections between bipolar cells and stable cells. So as we can see there on our array, the red ones are the bipolar cells that are connected to our staba cells. And by the information we found on this other paper, we discovered the amount of synapses that both cells realize, and it's about 0.39 synapses per micrometer. And basically, that's how we produced our connection, or our connection. Basically, we have on the 300 by 300 simulation area, we have 405 bipolar cells on average. 100 connect to the staba cells, and they made it an average 500 synapses. So this is one of the properties that we wanted to explore with this first experiment. Basically, we have established on the literature that this kind of circuit, it has a directional selectivity. Basically, it's a selectivity centrifugal. That means if a stimuli is coming from the dendrite to the soma, we have a reduced response, whereas with centrifugal from the soma to the dendrite, the response is larger. Here, we try to mimic this experiment, basically. And what we did, we had this ring, and this ring, it was, we programmed it to move on a centrifugal or a centripetal fashion. And then when it achieves our bipolar cells, basically it made it activates and makes the excitatory synapse. As we can find in our simulation data, we were able to reproduce this difference, this difference response. Here, basically, we had two velocities for those, for the contraction and the expansion of our ring. And basically, they found in experimental data about a delta V, a difference of about 3-4 millivolts, and we were able to achieve one. Here we have roughly 1 and 1.5 millivolts of difference. So it matches the experimental data. Next, we did the modeling of DSCGs, the DS cells. Here's the morphology of this cell. This cell actually, it spikes, different from the last one. And it has, well, basically that's the morphology. It also has those axons because the information that it receives, it goes through the cortex via the optical nerve through those axons, but we didn't include the axon in our morphology model. That's this one. To do those spikes, we use the HH-like mechanism that include sodium, potassium, and calcium that were responsible for our spiking behavior of our cell, and the HCN channels, that it's a channel gated by a protein. Basically, it is responsible for this behavior when the cell hyperpolarizes to regulate the hyperpolarization. So here is the experimental data taken from the response of this cell for constant currents from a lot of constant currents. And here is our simulation data. We were able to fit it. It had a really good result here. And basically, now we have to connect the bipolar cells to the SCG cells. What we did here is we used basically the same rules that we found before, but the only difference here is that the bipolar cells, it connects towards all, through all the dendrites. It doesn't have a special difference. So basically, that's how our array looks when we connect the bipolar cells to the DS cells.
Thank you. So now we start and have a look at what we have. We have a bipolar cell array, a simplified one. We have a realistic, detailed model for staba cell that connects to the bipolar cell array in a realistic manner and acts like a real one. We have a DS cell model, detailed one, and connect to the bipolar cell array and acts like a real one. So before we before I continue, next step would be combine them together to make the network. Before that, let's stop a little bit and have an estimation of the scale of the simulation. Basically, I told you that we have 90 Starburst Summer cells connect to one DSL. How do we know that? So we know the cell density of Starburst cell is like between 400 to 500 cells per millimeter square, and which is a smaller cell density, but considering the dendritic field is much larger, we have to allow a certain level of overlapping to reach this cell density. So we generate this regular hexagonal array, and by manipulating the overlap, the distance of overlapping, we're trying to reach this cell density. And that's we allow an overlap of 150 microns and reaching the cell density of 150. And things can increase there. That's an overlap of 190 microns, and the cell density is finally in this range. And next question is, so how many cells are there? The black circle in the middle is basically the size of the dendritic field of a DSL, and all the red ones are connected to it. We count it in the program, it's about 90. So I'm gonna jump this here and I'm gonna come back later. So 90 cells is too much for us. We don't have the computational power, I don't have the time, and remember we are simplified detailed model. So we start from one-to-one connection, one Starburst cell to one DSL. And also their connection is following realistic rules. Remember this plot? So basically the blue one tells us the bipolar cell input only stays in the proximal 2/3, and the black one is the Starburst cell output. It only happens in the distal 1/3, which happened there. The red dots are the synapses between Starburst summer cell and DSL. And from this model, we have a Starburst cell, we have DSL, we have 1,000 bipolar cells, 90 connected to Starburst cell, 140 connect to DSL, and there are like 160 intersections between them. And the bipolar cell makes about 500 synapses to Starburst cell and making 1,000 synapses to the DSL. And now we have a problem, because in the real world, in the real retina, there are about 90 Starburst cells, and in the real network, the excitatory input and inhibitory input well balanced. But here we only have one Starburst summer, which means our inhibition is way smaller than the real case. And we actually run with realistic parameters, and it happens that we cannot observe any inhibition because it's so weak and it's flooded by the extremely large amount of bipolar cells. So how are we gonna do? Should we give up? Nope. So we build our tempered, simplified detail model in a way that we change the parameter in an unrealistic way so that we can observe some network phenomena with this only one-to-one connection. The first thing we tempered with is the weight of the inhibitory synapses. You can see it's 100, which means 100 millisiemens, which is extremely large synapses, inhibitory synapses. So that is to simulate, because the cell normally received way more inhibition. So that is a simulation. What happens if we have a stronger inhibition? Can we observe some realistic network property if we do that? So actually, one characteristic property of the DS circuit is, as I mentioned before, it spikes a lot to the peripheral direction movement and spikes very few to the other direction. And can we replicate that? And yes, we do. So we project the light bar moving in 8 directions and counting the number of action potentials, and it showed this tuning. So it's pretty cool because it's only one-to-one connection, but tempered with, just forget about it. So at this time point, we have our very simplified model, and probably you forgot about why we do this, because we want to study the flash, the effect of the flash on this. So we try to do that. Also, we still have the problem of the bipolar cells because the excitation is too strong. So what we tempered with again is we remove the bipolar cell DSL connection, but keep the bipolar cell to Starburst cell connection. Another thing is we current injection to the soma of the DSL to simulate what happens if the cell is excited. And here it goes. So, okay, sorry. Here it goes. Thank you. Oh, thank you. So basically, when there's no flash, the Starburst cells stay still. The DSL, because of the current injection, have a tonic firing. And when there's a flash, the Starburst got activated and provided a prolonged inhibition regardless of the preferred direction or non-direction. So that's basically matches our hypothesis. And that's it. And okay, a long list, but I didn't put there. Thank you for your attention.
Yes, of course. So Arne wants to know how does direction selectivity of the starburst amacrine cell depend on the density of dendritic voltage-gated calcium, potassium, and K channels?
You mean the direction selectivity on the starburst cells?
Yes. For example, when you show the centrifugal centripetal. Yes. Did you analyze how it changes depending on the density of dendritic?
The answer is, sorry, no, not yet. But it's good. It's an interesting question. So currently we just use some default parameters for all the channels we found. And actually, it's still not a very realistic model because there are still some channels we didn't include it in the model. And basically this simulation tells us how difficult it could be to get from simulation to the real world. It's like there's a sentence saying like the most accurate map to a city is the city itself, but it's useless as a map. So actually, every kind of simulation is a simplification of the real world, but then it's very important how we interpret our result and how we do the simplification as here. So basically, I guess I'm not answering question, but yeah, no, sorry.
And just a comment from Arndt, he said that the direction tuning results are very nice.
Thank you. I think it's very nice as well. Thank you. Yes?
I like a lot your job. Congratulations. So this experiment was from NASA, and it was, well, these results kind of trying to mimic why the flash stops these bad feelings. So at the end, you reproduce that this is due to the hyperpolarization that you get?
Yes, that's what we think. Actually, I didn't have time to explain my hypothesis. I think it's something like a Doppler effect because I mean, it's too complete. Yes, but it's because of the prolonged inhibition or prolonged hyperpolarization from the starburst cell get like regularly activated, and that is basically regardless of the direction. So basically, what happened is like all DS cells are inhibited, and they provide no direction information to the brain. So therefore, there's no conflict, and that's our hypothesis.
Great, thank you.
Thank you.
Fast question.
Did you work on this type of modeling before?
No.
Or you started everything here in LASCON?
I didn't know neuron before this, but
okay.
I know.
Not you, but all of you, no?
Yes. Yeah.
Good. Okay, so let's proceed to the next.
Thank you.
Hello, my name is Marina, and this is Ricardo, and we're going to present our project about the comparison between reduced and realistic models of motor neurons morphology and mechanisms. So this is a brief outline of what we will present, and I will begin with context, objectives, and the beginning of methods, and then Ricardo will continue with methods, results, and conclusion. So, a brief context, motor neuron activation is the final step in movement control, and their input-output properties are highly nonlinear. And this behavior is mainly due to the activation of persistent inward current, also known as peaks, mediated by voltage-gated sodium and calcium channels in the dendrites of motor neurons. So, persistent inward currents are present in many types of neurons, but in spinal motor neurons, they are especially strong. And they are present mostly in the dendritic region of the motor neuron. And also they are subject to a strong neuromodulation of monoamines, such as norepinephrine and what's the other one? Serotonin, yeah. Okay. And this is to show the effects of the monoaminergic input. And here we have the, as you can see, peak. The peak is highly dependent of this monoaminergic input. As we can see here, in the green line, we have this high level of neuromodulatory input, and we have the strongest persistent inward current. And as soon as it reaches like above minus 40 millivolts, the synaptic input decreases like dramatically. And this dependence of the peak in the neuromodulatory, of the monoaminergic neuromodulatory input on the peak can be seen also in this graph of the muscle force. As you can see here, the highest level of the neuromodulatory input, the highest is the gain and the muscle force. So it is needed less synaptic input to reach the maximum muscle force. Okay. Now I will present some of the characteristics of some of the properties of the persistent inward current. And one of the basic properties is that when subject to this ramp voltage comment, the peak represents this complex behavior that can be seen here, that once the voltage is linearly increasing, you see that has this downward deflection. Also, when it's linearly increasing, you also have this deflection. And we can see here also in this plot, when we have this deflection, that marks the begin of the persistent inward current, and also this second deflection that marks the end of the persistent inward current. And one of the characteristics of this peak is that it presents this hysteresis that can be measured by the difference between the onset and offset points. And also this hysteresis is present in this relation of the firing rate and injected current, as we can see here, both in the experimental result and the simulated model. And another property of the persistent inward current is that it is this prolongation of synaptic input. As we can see here, there's the short input, and then the cells starts firing, and there's the peak amplification. And once the input is turned down, the cells continues to fire, and the peak maintains. So this phenomenon is important. It helps in the maintenance of posture, for example. So with that said, in the literature, it's said that it's kind of difficult to represent this phenomenon in simplified motor neuron models, like the one with only 2 compartments. So our proposal is to model for 2 different approaches of motor neurons, one more complex and one more simple, to try to reproduce this electrophysiological phenomenon. So we'll be modeling a 4-dendritic compartment that is based on this model of Powers and Heckman. And then we'll perform a reduction and reparametrization of the model to try to represent the electrophysiological phenomenon. So to do this, we basically had this original model that so it had 4 dendrites, and we changed it to the 2-compartment model by maintaining the same dendritic membrane area. And also in the original model, we had this variability of the calcium channel conductance, that it was to maintain the results as close as possible with the experimental results. And then we tuned this conductance by adjusting it to what would fit better to represent as close as possible the results. So here is a passive model analysis of how it was done. So we injected this alternating current to the soma and measured on the dendrite, and then we did the other way. So the impedance had to be the same. And to verify if our model was correctly reparameterized, we measured on the two models. And as we can see here in this curves that present the impedance of the dendrite, the stimulus on the dendrite and the measure on the soma, and the stimulus on the soma and the measured on the dendrite, they got almost the same results for both the one dendrite passive model and the four dendrite passive model.
Hello, good morning. Now we're gonna play with some ionic channels. We are showing some results, impedance results of the passive model, just capacitance and membrane resistance. And we inserted these mechanisms to represent the actual potentials. There is persistent and transient sodium channels, the delayed rectifier potassium channel, and we added some, these are on the soma. Also on the soma, we added these after hyperpolarization channels, and on the dendrite, we added these persistent more current channels that represent most of the nonlinear properties of the mononeurons. And we made the same analysis that Marina showed us with these properties, and we can see here the difference is that we made a current clamp at the soma, and with these currents we are showing here. Sorry about the confusion of but the colors are the same for the same current, and we can see on the discontinued line, it's the impedance at the soma, and the dashed line here is on the dendrite. They are quite similar. There is a little bit of difference here on the dendrite, we can see, but we think that was okay. And we continue with the analysis. So now here we made a triangular current clamp to the soma, and we can see the results here for both models, and the left one for the one dendrite. The first graph at the top is the firing rate. We can see it's quite similar. Here's the membrane potential at dendrites, at the soma. They are also quite similar. We can see here the facilitation here on both models. And okay, I think that's it. Okay, this is important. In both models, we could represent the firing rate in hysteresis. And these are experimental results with CAT, with a median level of neuromodulatory. And at the end, in the conclusion and discussion, I will show one hypothesis that are based on this. So remember this difference on the descending curve. This is the relation of the firing rate over the injected current of the current clamp. So this is the ascending phase. We can see the first phase, the second phase of the different gains, right, for that are caused by the persistent word currents peaks, and there's a little difference on the descending part of this graph. So on in the next, so here we made a voltage clamp at the soma on both models, and we can see the membrane potential. On the first graph, the MCA is the activation variable of the L-type calcium channels on the dendrite. We can see here also the clamp current. So we can see it's quite clear here the onset of when this activation variable is high. We see here the downward of the current clamp. Both models represent very good these. They are quite similar. There's a little bit difference here in this star case-like current on the descending part, but like here on the experimental results, but they represent the hysteresis on the difference. The hysteresis calculated by the difference of the onset of the peak. This is the ascending curve, and the descending curve here is the offset when this here, the descending curve, right? So both models could represent this these nonlinear features of model neuron. This is quite a striking result. And we made some analysis with combined oscillatory and inhibitory synapses. In 3 combinations, we made this, this green triangle represents the oscillatory synaptic input, and the red ones represent a constant one. This inverse triangle one, we're gonna say this is a push-pull model of station, and this one is a balanced one. And we can see the firing rate of the model neuron changes depending on this kind of station. And we can see the firing rate here, membrane potential, and the activation of the variable of the calcium channel on the dendrite. And this here is the constant inhibition. So we can see here in orange the median value of the synaptic current. This is a stochastic process, so we show it the real value here in blue. In the green one is the inhibition. So they are quite similar. There is a little more variability on the 2-compartmental model, and we made an analysis. We made an histogram analysis of the interspike interval. And the mean value, we made 10 simulations because of the stochastic process. We have a more realistic value of the representation of the model. 10 simulations of 12 seconds, and we got these results. The mean value of the histogram is quite similar, and the variability also. And now we made the simulation with the push-pull input, where you can see here, the starts with a higher inhibition, go to 0, and go higher again, like an inverse triangle. And they are the same plots, but different results. We see this more triangle shape of the firing rate curve, and more variability at the 2-compartment model. And this is the interspike histogram. And we can see the mean value is quite similar also. The variability also is quite similar. And this graph here shows for a balanced input. So we have these 2 triangle inputs. The inhibition is quite a little lower than the excitation. So they have quite similar response for the stimuli. And they actually shows a phenomenon here that the B stability, they don't stop firing for this conductance, the activation channel of the L-type response for the peak keeps sustaining the fire, even if we remove the input. And we made a histogram of that also, and here it is. We have some changes in variability of both models. And we are not sure, but we hypothesize that this is because of this difference of the descending curve, that at the 2-compartment models, he keeps firing for lower rates, for lower values of injected current. And usually we see at lower firing rates a higher variability in motor neurons. Maybe this is the cause of this difference. We're not sure, we need more tests. But in conclusion, we could represent the nonlinearities of the model, like BT, the peak hysteresis, the firing rate hysteresis, facilitation and saturation. And this kind of difference that you saw on the firing rate, we are not sure yet, but we hypothesize that this can be the cause, this difference on the firing rate on the descending curve. And that's it. We thank you for your attention, and our sponsors, Neuromat, for having us. And questions?
We have two questions from YouTube. One is from Bill Litton. He wants to know what neuron was used on the experimental data. It's
It
asymmetry
is
neuron?
a spinal cord motor neuron of a cat.
Yes.
We have anesthetized. It is an alpha motor neuron from cat. There is, I think there are in many states, depending on the state of the motor neuron, it represents different features, like on this figure here, where we show on the left graph, we have 3 states, like low, medium, and high-level neuromodulation. So most of the results are alpha motor neuron of cats.
And now a comment from Arndt and a question. He said he liked the strategy to match the transfer impedance between the two dendrites model, the two models. And the question is the difference you saw in the hysteresis. So does that reflect the heterogeneity of excitability in the four dendrite versus the lack of heterogeneity in the simple dendrite model?
I'm not sure if I understand the question, because most of the, can you put the
There was a slight difference on the hysteresis, right?
Yeah, there's a little difference. I think this is because we defined different conductance from the L channel on the dendrite, and the star case like here perhaps is because each dendrite activates and saturates at a specific time. So I think that's explained. I'm not sure if I responded the question.
Probably it's because each calcium channel has a different conductance, I guess.
Oh, yes. So I think, yes, that would be the answer. And from Leonardo Abdallah Elias has also a question. How the spike train variability was calculated in the triangular simulation?
We calculated for each simulation, we calculated the interspike interval, and then we concatenate all the simulations, all the intervals, and we calculated the mean, the standard deviation divided by the mean, and that's it. That's how we calculate the mean and the variability of the interspikes.
Okay. Well, we are Paula and Evelina. We are presented our project of the LASCON School that is centered in one of the most important behaviorals in neuroscience, that is the fear conditioning. The fear is very important for the survival of the individuals of the organism because they have to learn to associate two kinds of stimulus, one that is neutral and one that is fear. So fear conditioning is one of the paradigms widely used in neuroscience. It consists in that the animal have to learn to associate a neutral stimulus, that's the tone, or to another stimulus, that is the conditional stimulus, that is the shock. When the animal match these both stimulus, they learn to emit a kind of responses that will be important for the survival. The network of this kind of, the network of fear conditioning is very, very studied, and there are a lot of regions that are involved in them, as prefrontal cortex, hippocampus, and the amygdala. So we are interested in this particular network, and for that we have these objectives. That is that the stimulate a fear conditioning network composed of these three brain regions that are the amygdala, prefrontal cortex, and hippocampus. And because of when the stimulus is very, very strong, sometimes this behavior, instead of be adaptive, this is an adaptive behavior. So we want to know what happens when the stimulus is very high, and for that we simulate this network with different intensities of the stimulus in order to see the plasticity and the synaptic change in each population. Our hypothesis is that the higher the intensity of the stimulus, the higher the response, even when we distort the connectivity of the network. To prove this hypothesis, we plan this methodology. We use cell templates from the model DB, and we incorporate this cell templates with Python and NetPine. We use our principal reference with that from Kim and collaborators. And at the beginning, we want to incorporate the cell templates for the regions that we were interested, hippocampus, amygdala, and prefrontal cortex, but we have a little bit of troubles when we want to incorporate them. And also the network between all these regions is very, very, very complex, so we only stay with the amygdala, in particular the lateral amygdala, that is that one that Kim and collaborators works with it. This is the network that is proposed by these authors. So the lateral amygdala have pyramidal neurons and interneurons. The pyramidal neurons are classified by three other kinds of neurons, depending of their firing characteristics. So we have the pyramidal neurons type A, B, and C, and a population of interneurons. It is well known that not all the neurons receive the same kind of stimulus when they are in the field conditioning in alive animals. So we perform this, by the training, we perform these different stimulus in different kind of populations. So in the case of the conditioning training, we administrate a tone and a shock. We first administer the tone, and just 100 milliseconds before the tone is ended, we put the shock. The tone was in a frequency of 70 hertz, and the shock in a frequency of 200 hertz. So some neurons of the type, the pyramidal, apyramidal neuron receive the tone. A half of neurons of the pyramidal B receive also the tone, and all the neurons of the C pyramidals receive also the tone. For the shock, the 60% of the pyramidals, apyramidals receive the shock, and only the 50% of the pyramidals, B pyramidals receive also the shock, and the interneurons both receive the stimulus of the shock. So Paula will gonna talk a little bit more about this network.
We tried to perform the same network at the KIMS. So the KIMS have this kind of topology. It has a kind of horn shape, and there's separated part for the dorsal part and the ventral part of the amygdala, and also there are interneurons among the cells. But we didn't use this kind of topology because it's too hard, and we only created the usual. I will show it later. And then we have used the cells that are provided in the KIMS project. So there are this kind of conductance. We didn't change anything because we wanted to see the whole network. And then we used the kind of proportion that was given in the paper. So the proportion between pyramidal cells and interneurons was 80 to 20. And only for the pyramidal cells, they are also divided in some proportions. A was 50, and then type C was 30, and type C was 20% of that kind of proportion. And to modulate, you use it, in the KIMS, they used a total number of cells of 1,000, but because of problems in our computation, we used only 100 cells, and so half of the cells was related to the dorsal part, and the other half related to the ventral part, and used all of the cells, type A, B, and C, the interneurons, and to simulate the background noise. He is related to the other kind of regions that this network is connected. We used a Poisson with the rate of 70 because this was, we tried empirically to create the same kind of behavior that was written in the paper. So we used this number of synaptic mechanisms that was given in the paper. And the synaptic weights, we have to change a little bit because of the size of the network. And we created a population for the dorsal part, other for the ventral part with the A, B, and C interconnected. And the interneurons, they are interconnected between dorsal and ventral, they are connected among themselves. And we created this separately because the stimulus was applied only in the dorsal part. So we think that because of this, they put the same, they separated in the paper. So this is the cell model that we downloaded from ModelDB. I think this is like type B, but they are not so different among themselves like in the picture. And before creating the network, we try to see that the cells are really working. So we put a current at the same type of current that they're showing in the paper. And we could see that applying this current, the type A behaved the same way that it was shown in the paper, and as the same to type B, C, and the interneurons. And only putting the background noise, but without any connection, we could see that the network was firing. Here is related to the dorsal part. Here is related to the ventral part and the interneurons in the back. You can see that without any kind of stimulus, they are firing more or less at the same rate. And then when we put shock, but they're still not connected, we can see that they're not connections, only the shock in the
dorsal part, we see that they are firing in the same, they are firing only the cells that the current was applied. And the tone, the same thing, only the cells that received the current had some kind of activity. And then we put the connections. The blue ones was a inhibitory, and the red ones was oscillatory. And we could see that with the connections, other kinds of cells were more active because of the connections.
Okay. Here we put set the network, and we administrate the shock. Here is in yellow is the time where we put the shock, and we can see the interneurons response in with more frequency, and also the ventral part. This is the ventral part, and because these have connection between this is the dorsal part, and this is the ventral. Because these have connection with them, they begin to fire almost in the same time. Here are the firing rate. We can see that the interneurons again is the one that more responds to this kind of stimulus. Remember that the stimulus is set to 200 Hertz. In the case of the tone, the response is a little bit different, also because the tone is set in a more time window, and we can see that the interneurons doesn't speak higher as in the previous case. In this case, also when they receive the stimulus of the tone, they don't fire at the same time. In this both case that I show, the network are connected, but we don't follow the proportion that Kim's proposed. So here we set with the proportions of each cell, and we can see that the firing of all the cells are not less. They spike less. And the interneurons, all again, we can see that they spikes with a high frequency and with more synchrony. And here is the same case for the tone with the proportion of the cells that Kim proposed, and we can see that there are not at this time we cannot see a kind of plasticity because they don't have a relationship between the tone and the shock, and there are only the conditional or the unconditional stimulus. And in this case, we perform a kind of conditioning. Here is the tone, followed by the shock, and we can see that they begins to respond to the tone at the first time at the tone, and later for the shock. And at this time, there are kind of synchrony between in the other regions, that is the dorsal part and the ventral part of the lateral amygdala. This is only a resume of the firing. We can see that the cells that respond higher are the interneurons, and no, it doesn't occur for the other kind of pyramidal neurons. And when we administer the tone and the shock, it's only to see that the rate here is to 10, and when we put both the stimulus, the rate is higher. And when we try to put the plasticity, plasticity mechanism, we didn't see any difference when in this plot we put the plasticity mechanism, and this is with the plasticity, and we see the same, it's the same figure. So in this case, we were not able to reproduce the plasticity it was a little bit difficult to read the parameters that the authors use for the plasticity. They use a Hebbian plasticity, and it's something that we have to work in order to look what happened. So we can see plasticity, and the conclusion is that we could simulate the lateral amygdala network, and there was differences between the responses with the shock and with the tone, and also that the number of cells that receive the stimulus changed totally the network. So it's very important to take in account the proportion of the cells that respond to each stimulus, and we could not see the plasticity in the model. And the bibliography and the acknowledgement. Thank you.
Não sei se é comentário ou pergunta.
So your model is relatively complex in the sense that it includes many different cell types. So did you get some understanding out of your modeling of the roles that the differences between the cell types play in shaping the network behavior? Because one of us could the most prominent effects in your results were due to the connections. In principle, do you think that you could have got similar results by dropping the cellular diversity?
I think we'll not have the same because the final rate is different. Because we can see in that auto plot that type C fires more. So if we put the same current, the type C would spike more than the other. So I think that because of these different chains, it will not be the same behavior.
But you could have taken just the same point neurons with a different excitability, for instance, or with a different resistance. This would have given different firing rates. So is it a pure firing rate effect, or the actual detailed nonlinear firing and adapting behavior matter?
We couldn't see this now because we have some problems to simulate the network. So this we wanted to test without all the cells to see that it really matters, but we couldn't do this because of the time. So we can't see like for sure that maybe we only know that because of the fire rate it will be different, but we don't check really.
So from Salvador, these are his words, very nice work. And he wants to know how did you set up the connection weights and probabilities? Did you need to tune these values until you saw some reasonable spiking?
We try to change all these parameters, the weight and the probabilities, but we set until we found the activities that in the Kim's paper show. But yes, we play with these parameters and we found another kind of things. Sometimes they didn't spike when we changed the parameter, the weights, and also we set the probabilities between when we input the excitatory inputs and the inhibitory inputs.
How do you put this in?
Good
morning to everyone. My name is Bruno Barreto. I'm doing my PhD in physics in Universidade Federal do Paraná. And
Hi, my name is Cindy. I come from the University, National Autonomous University of Mexico. And we are going to present our project for LASCON, which is titled Modeling of the Chloride Current Carried by GABA Receptor in Oligodendrocyte Precursor Cells. Can you help me with this? Okay, so the objective for our project was to model in silico this current that you can record from a glial cell, and this occurs through the flow of chloride ions in this receptor, which is the GABA receptor. It's the GABA receptor because it only opens when the GABA binds to this. So why we choose oligodendrocyte precursor cells? Well, first, let me introduce to you that the composition in our brain is, of course, we have a lot of neurons, but an equal number also of glial cells. Among the glial cells, there are several types of glial cells, and they are also called non-excitable cells because they don't have the action potential. But that doesn't mean that they are not excitable, because when you put an electrode and you measure the difference in voltage membrane with respect to the extracellular space, you realize that all cells have a negative resting membrane potential. And also for other studies, for example, with calcium fluorescence, you can see dynamics in these cells. As our partners previously presented some calcium dynamics in astrocytes. Oligodendrocytes are responsible to produce the myelin that covers the axon in neurons. And this myelin is very important because it increases the resistance of the membrane of the neuron. So this is very important for the velocity of the action potential. When we have a mature brain, the myelin is still constantly remodeling because this helps to have plasticity in our brain, so we can constantly regulate by learning the velocity of action potential when needed. So at the end, this will have an effect on networks. So this myelin is affected in pathologies such as multiple sclerosis, so it is very important to study this type of cell. But before we have a mature oligodendrocyte, we have some other stages, previous stages that drive into mature oligodendrocyte. These are called the oligodendrocyte precursor cells. So from now on, we are gonna call these cells OPCs, oligodendrocyte precursor cells. So when we born, we have a high population of these progenitors that are bipolar. But as the oligodendrocyte matures, we see that we have more complexity. And in the adult brain, we have a high density of these cells. And so even though we are now adults and we have many paths already myelinated, we have a lot of OPCs present in our brain. And they are very important to us because in case that we suffer some pathology that causes that we lost myelin, they are present there, so we need them to generate new myelin. And they are also important because they have been described to receive synapses from neurons. And why do they receive synapses? Because they have the receptors to receive this signal. So they have a lot of receptors for a lot of different neurotransmitters and a lot of ionic receptors and metabotropic receptors. So why we choose a chloride current? Well, we choose a chloride current because, can you put? Yeah, okay, can you hear me? Okay, we choose a chloride current because the activation of GABA receptors has been implicated to help these oligodendrocyte precursor cells to become mature oligodendrocytes. This means myelinating oligodendrocytes. So have you heard previously about the GABA receptor? So in neurons, for example, which are very popular, when they activate, what happens to neurons? Somebody who can help me with this information? When GABA receptors are activated in neurons, what usually happens? Inhibition. So you mostly know these receptors because they carry a chloride current, and chloride current is made up of negative ions. So the current that goes through these receptors usually hyperpolarized cells
because the current flows this way, and so negative charge enters the cell. But in the case of oligodendrocyte precursor cells, when the GABA receptor is activated by the union of a GABA molecule, the chloride flows the other way. So if we have a negative resting membrane potential, and now negative ions flows out of the cell, what happened? Somebody? Negative charge flowing out of a cell. What happens with the membrane? Resting membrane potential? Depolarized. Okay, thank you. So as you can see, is studying this current in another cell, which is a very important cell, is important so we can in the future understand better the system and understand better the implications of the activation of this chloride current that has also been implicated in other very specific physiology processes. And why flows the other way? Well, because the concentration of the chloride ions is different in this cell than in neurons. And as you can see, for the Nernst equation, there's this chemical gradient implicated in the way that this current has to flow. So before Bruno presents some of our results, I'm gonna briefly explain what are the components of the current that we study. So this is a culture. Here you can see several oligodendrocyte precursor cells, and this shiny stuff is a pipette. So because we are looking this through a microscope, we see this so shiny. But for you to understand how we record the current, well, this will be the pipette. Inside we have an electrode that records the current that flows through the channels that are present in the membrane of the cell. So when we record a current stimulated by a GABA, what we see is, okay, we have the first condition when we are just measuring the current. Since we are not opening any channel, we have the basal conductance. So in this case, here are the GABA receptors, but we don't have GABA molecules. The blue dots will be chloride ions. This will be the outside of the cell, and this is the inside of the cell. So at the beginning, we first record this area, but now that we have these yellow dots that are supposed to be the GABA stimulus, this GABA stimulus binds to GABA receptors, so GABA receptors open. So what we record now is a current that flows through these channels. So this is the current. So now we have a phenomenon in this current which is called desensitization. And desensitization means that even though we still have the GABA here outside the cell binding the receptor stops slowly to have this current through the membrane, because as the word says, desensitization, it loses the ability to be open in the presence of the only of one of the molecules that opens this receptor. So at the end, as in regular synapses, the chemicals must be watched out of the exterior of the cell. So we again have the basal condition when we don't have any GABA outside the cell and we don't have a current. So now we are back to the resting state.
So, all right. So how to model this current? To model this current, we need to look in the experimental data again. It's like this. But I will just turn the upside down and normalize this data to understand the shape of this curve here. So first, we thought that we can reproduce these results with an alpha synapse, an alpha function, but doesn't work. Then we found in the literature, in a paper that I showed to you, and in one book there, and we found that we can reproduce this behavior with a double exponential function, which is basically the multiplication of two functions. So the first exponential takes the contribution of the rising here, is the red, is the green line, and the blue in the other exponential is the decayment function. And the multiplication of these two functions is equal to this red line, which fits really good our data. So our challenge was to reproduce this on neuron. I don't know if you remember, but a long time ago, in the first week of this LASCON. Newton, Professor Arndt, and the other tutors show us the neuron program. And we found this paper where the authors uploaded the morphology of an OPC. And we need to learn how can we use that and how can we reproduce that desensitization behavior. This is the morphology of the cell, so this is part of the code. So let's see if it's work. Yeah. So here we need to load the morphology. And in this data, they put the morphology and the geometry, but they don't put the mechanism of the cell. And as Cindy told to us that this kind of cell doesn't have active potential, but have depolarization. So we make that, we insert a passive mechanism in our cell, and a passive mechanism with a very negative resting potential because of the chloride. And then we apply a double exponential function in the soma to see, to try to reproduce. The double exponential function is given by this equation here. And the conductance depends on our weight, which is the maximum conductance. And we have here the double exponential function. So let's see. This is an example of, in this paper, this is an example of how they study the GABA currents in the OPCs as a function of the concentration of the GABA. And we can saw here that for higher is the concentration of the GABA, higher is the amplitude of the current. And the amplitude of the current as a function of the concentration of GABA evolves as the sigmoid shape as a logarithmic here. So now our results. Here we have the application of the double exponential function in the soma, but in this case we don't have the concentration of the GABA. In this case we study as a function of the weight of the current. So in the first panel we have the depolarization is the membrane potential of the cell as a function of time, and the second panel here we have the current, the applied current. And we also study the amplitude of this current as a function of the weight. And here we also reproduce that sigmoid shape as in a log scale. We also made the application, instead of application applies only in the double exponential function in the soma, we also make the application in one of the processes of the cell. So the result here again is the membrane potential, the applied current, and again, we can reproduce that sigmoid shape here. And the cell always also depolarized in this case. And to compare these two cases, when we apply only in the soma and both in the soma and a process, when we apply the current also in the process, the amplitude of the current increase faster than the other case. This is why this happens because the soma are receiving the signal of the process also. Our final consideration. So the outward current of chloride can be simulated using a double exponential function. The amplitude of the outer current evolves as a sigmoid function of the concentration. And using neuron, we are able to reproduce this behavior with the application of an external current. Thank you, people of organization, the tutors, which help us a lot, Professor Arn also, and thank you.
So we have a question from Bill Lipton. He wants to know if the results of the intracellular chloride concentration is from culture or from slice.
In the case of the experimental data that we are citing, it was from an article using a cell culture. But of course, when you are using a pad clamp techniques, you are controlling the concentration of chlorides inside and outside. However, it has been shown also in vivo that the chloride in this type of cells is also has a different concentration than neurons, and the current is a depolarizing current.
Yes, and I think Bill also mentioned about the data probably you're talking about. You mentioned differences in the difference of chloride concentration, even outside the difference between neurons.
Yes,
right.
The difference with the glial cells, because also astrocytes have this property, is that inside the cell they have a little bit more micromolar concentration of chloride. It's a little bit. If you play with the concentration of chloride outside and inside, you will move the equilibrium potential for this current. So we found a very interesting paper that talks about what will happen in processes, because processes are very small. So in there, very fast, the concentration of ions will be different outside and inside due to the activation of GABA receptors. So it will be very interesting not also to model how is the diffusion of chloride ions inside these cells because of its structure, but also to study what will happen if we have a constant input or stimulation of these receptors, and that we change the equilibrium potential for the chloride current in the process. So instead, for example, in this cell, we will now have a hyperpolarizing effect on processes, but if you study this in neurons, you can revert also the equilibrium potential, so you will have a depolarizing effect in neurons. So that is very interesting to study.
Do you have any idea what is the difference in extracellular chloride concentration in white matter and gray matter? I think there's a note about Staley.
No, I don't have that information.
Okay.
So I'm not sure I did get it right. When you say that you're injecting a current, you're activating a conductance. Is that right?
Yeah, conductance is the weight of the conductance.
Okay, so and you're just looking at the loss of a clamp, no? I mean, you're simulating a voltage clamp condition or a current clamp condition?
No, we don't use the voltage clamp condition in this data here.
Okay, so but you're saying the fact that the larger the conductance, the stronger the depolarization, the smaller the driving force. And therefore you have saturation. That has nothing to do with the saturating effect of GABA on the ionotropic receptor. And by the way, there's two GABA molecules that have to be bound to the channel to open it, not one.
Mm-hmm.
The thing is that we are using the conductance as equivalence of the concentration that would be in the extracellular space, because we couldn't control. We were just starting to play with this, so we couldn't control also the duration of the application, which is also very important. So we just play with the conductance. But in this case, first we also tried with putting a voltage clamp in this cell, and we saw that the behavior of the concentration versus the amplitude of the current was not as from experimental data. So after removing the clamp, we saw that it was more similar. But of course, the conductance would depend also on the number of GABA receptors activated. And in this case, we're only applying one. And when we apply two in the dendrite and in the soma, we see the sum of these currents. But of course, we will need to also know the density of the receptors that are present also in the soma, and if there are differences with the number of receptors expressed in the processes. But this is, of course, we will need to work, we are planning to work more on this, so we can also include other parameters, other conductances, because the membrane also activates voltage-gated channels, so it's very important.
So, good morning, you all. My name is Raul, and this is my colleague May, and we are going to present our LASCON project, which is entitled Simulation of Glutamate Dysfunction on Single Frontal Network. And here's a brief outline of our presentation. First, we are going to give our motivation, then the model, the numerical results, and finally the conclusions. So, why the motivation? Why we are trying to model the singular frontal cortex? Well, the singular frontal cortex is responsible for processing sensory, motor, cognitive inputs, emotional and information and process, and also he deals with learning and we are interested in two regions specifically, the anterior cingulate cortex and the dorsolateral prefrontal cortex. And the ventral anterior cortex is located like around here, and the dorsolateral cortex is around here. So, first, the dorsolateral prefrontal cortex. The dorsolateral prefrontal cortex deals with cognitive processes such as working memory, action selection, but it's not always like, sometimes it deals with the memory, the action selection without working with the memory component. But the role of the dorsolateral prefrontal cortex is like, it don't deal with these tasks alone. Studies suggest that the ventral lateral prefrontal region also perform these working memory tasks. And the anterior cingulate cortex, it deals with the error detection, decision-making, motivation, and modulation of emotional responses. And some studies suggest that when we try to look to the cingulate cortex, studies suggest that the anterior cingulate cortex is like a hub of this network, and it shows like a really important role in the symptoms of major depression disorder. And I think that you all know depression, and it's the most common of all psychiatric disorders. And one of the symptoms can be like deficits in cognitive, circadian, and motor functions. And when we try to, and some neuroimaging studies reveals that it can present hyperactivity in the VACC and also hypoactivity in the dorsolateral prefrontal cortex. And also some patients can show like a lack of ability to switch from cognitive tasks and emotional tasks. And to try to simulate these symptoms, we based our work in this paper. And now I'm going to present the network, the model of the single frontal network. So we try to modulate a two-layer network, and in which layer we have 800 excitatory neurons and 200 inhibitory neurons. And all the neurons in the excitatory population are connected with each other, and also the inhibitory neurons. And the excitatory neurons are connected with the inhibitory neurons in the same layer and across the layers. And sorry. And each one of these networks receive external inputs, and the DLPFC network receives inputs from the posterior parietal cortex, which demands like cognitive tasks, and the VACC network receives inputs from the limbic system, which demands like emotional tasks. And to simulate the dynamics of the neurons, we use an integrated model where the currents, we have to consider the AMPA receptors, NMDA receptors, and the GABA receptors. And I think that you all know that the AMPA and the NMDA receptors are for oscillatory synapses, and the GABA receptor are for inhibitory synapses. And here you can see that we have to consider the AMPA inputs from the neurons and from the external inputs. And each one of these currents were modulated by this equation right here, where this S is the gating variable and the G is the conductance. And for the AMPA and the GABA currents, we have to deal only with this part. So it's like a exponential with a decay. And for the NMAD, we have to consider this part here, where the X is the neurotransmitter concentration in the membrane. And we also consider that the NMDA conductance was voltage dependent, so we have to add this part right here. And to simulate the MDD symptoms that I was talking about, we have to simulate the glutamate decay, a deficit in the glutamate decay uptake by increasing the time constant of the decay of the AMPA receptor. So here is the values, like for the mild depression, we have 2.05, and for the severe, we are increasing, and then in the severe case, we have like 2.15 milliseconds of characteristic time. And now May is going to present some numerical results.
Okay, so our objective from simulating these results is to see how the depression could change the dynamics between the VECC network and the DLBFC network, and how the transition from emotion processing to cognitive processing could be changed under the case of depression, and the role of glutamate in this case. So to simulate this, we designed a task for 5 seconds. And these 5 seconds, we included emotional tasks and cognition tasks, and that's how we designed it. That's the 5 seconds. So first 1.25 seconds, it's for just resting state, just noise, pulses, spike trains, with no emotional stimulation or cognition stimulation. And then this was followed by emotional stimulation in the VECC network. It's for sadness provocation. So it's like we are stimulating sadness. And we included 3 stimulus. Each stimulus was for 250 millisecond, and 250 millisecond difference between each stimulus. Then we shifted from the stimulation of emotional task to stimulation of cognition task, which is a stimulation in the DLBFC network for working memory, and it's the same stimulus with the same duration. And after that, the last 1.25 seconds, it's just for resting to see how the networks will perform after stopping the stimulation. So that's the design of our tasks. We stimulated 4 cases, which is the first one is the healthy case with no depression. And the 3 other cases is for the 3 different cases depressions, or representing cases depressions, which is mild, moderate, and severe. And as Hau said, that it's just differences in the slowdown in the glutamate decay. So now we will show our results. This is the simulation of the healthy case. This is the raster plots of the 2 networks, and this is the average population rate of the 2 networks. The blue one is for the VECC, and the red one is the DLBFC. So what we can see here is that starting from 0 to around 1.25 seconds, the 2 networks are in resting state, no firing, nearly no firing, no firing. And when we started stimulating the emotional task here the VECC started to fire, so because it's stimulated, and the firing increased to this level. And then when we started to stimulate the DLBFC network, the working memory network, the VECC started to decay. And then after ending the stimulation of the DLBFC network, the firing rate of the DLBFC network, it continued, which means it has persistent activity. So what this figure says about the healthy case is that healthy people could do transition from emotional stimulation to cognition stimulation, and they can have persistent activity in working memory tasks. So what's the case in the depression? For the mild depression, the main difference we can see is that here the VACC network, the emotional network, it started to fire even without stimulation, which means that there is emotional fire, which can represent negative thoughts even without emotional stimulation. That's for the mild one. But the thing is, when we stimulated the DLBFC network, the VACC, it decayed, which is good. It's just mild depression. Then for the moderate case, what we can see is that persistent firing here, it's still the same, but it's earlier. And here, the VACC network, it nearly didn't stop. It still has firing, even in the resting state and after stimulating the DLBFC network. So it's like the negative thoughts are still there. For the severe case, the thing is the VACC doesn't stop and the DLBFC started to decay. So the cognition tasks started to decay. Just to compare these results, this is the VACC network, and we can see that here, the healthy case, how the firing of the depression case is almost in resting state, and here for the moderate and the severe case, the VACC doesn't stop. So that's the hyperactivity in the VACC. Then we needed to stimulate some treatments for depression. So for the paper we are implementing, it's stimulating the SSRI treatment, and we tried to test another treatment which is representing the NMDA inhibition, which is the NMDA receptor antagonist. And we just decreased the synaptic conductance by 10% factors to see the difference. So that's the figure that shows the healthy and the SSRI and the NMDA receptor antagonist results. So we can see that for the mild case, the SSRI and the NMDA receptor antagonist, they are nearly close, but the firing is still, there is some firing here in the resting state, but they were able to go down after the stimulation of the DLBFC. And we can play with the factors of the dose of the SSRI and the NMDA antagonist. And finally, this is for the severe case. So for the severe case, as we remember that for the depression case, the VACC was always firing even after the stimulation of the DLBFC. But after the treatments, this doesn't happen. It decayed. So this says that the SSRI and the NMDA receptor antagonist could give some solutions for depression. And now, Haou will continue with the synchronization.
Yeah, and we also performed synchronization analysis in the networks. Specifically, we want to see like how the oscillatory populations or the synchronization about the oscillatory populations. And to perform this analysis, we use the Kuramoto order parameter. And the Kuramoto order parameter, we had to associate a phase to the membrane potential. And this is the equation that we use to perform the analysis, where these R is the Kuramoto order parameter, and it goes from 0, where is no synchronization, to 1, where is a lot of synchronization, like total synchronization. And this theta is the phase associated to each neuron, and is the size of the population that we are analyzing. So for the healthy case, first we can see that the synchronization in the networks are like very high. So like the membrane potentials of the neurons are almost the same. And when we perform the tasks first in the VACC, we can see that the synchronization drops a little, and then after the task, it comes back to the old value. And the same, and for the DLBFC network, we have like the same behavior. And unfortunately, we didn't have the time to do like more simulations, because you can see that this is not the best plot, because there are a lot of fluctuations. We had to perform a lot. The best thing to do is like to perform a lot of simulations and then plot the mean value, but we didn't have the time. And for the severe case, one thing that we can see is that the VACC network, it doesn't change with the tasks. So somehow we were not able to like conclude what is these mechanisms that do this, but somehow the network is not reacting to the input, like the synchronization doesn't change. And the behavior in the DLBFC networks, like strange, and somehow it's like it drops, but after some time come back, like in the other plot, we couldn't see that the response was more clear to the input. So it shows that the synchronization, the synchronization of the network is showing a behavior that is not like very clear what is happening in the network. But you can see the difference between a network of a healthy person and a person with severe depression. So, to summarize our presentation, we used a model to describe the glutamate dysfunction in the single frontal network. And this model was able to reproduce the symptoms that the glutamate dysfunction can cause, such as hyperactivity in the VACC and the lack of ability to switch from emotional and cognitive processing, and the hypoactivity in the DLPFC. And both treatments, the SSRI and the NMD receptor antagonists, showed good results. And just remember that the SSRI was proposed in the paper, and the NMD was implemented by May in our work. And finally, we can conclude that the synchronization patterns is also affected by this glutamate dysfunction. So this is our main references. And I would like to thank you all for your attention, especially Professor Roque for putting this school together and making this all possible. All the tutors, professors, and all you guys for this amazing school, this amazing month, intense month. And that is, do you have questions about our work?
Hi, a nice presentation. So it's not completely clear to me how you evaluate the phase, because the neurons don't have a clearly periodic
they
or
don't have
oscillatory
a clear
behavior.
phase, so I took the values of the membrane potential and I normalize it and associate it to a phase, like from 0 to 2 pi. So the minimum value was like 0, and the maximum value, the spike value, is 2 pi.
Okay,
so it's like oscillation.
Okay, so in reality, what your index is measuring more than a synchronization could also be a variation of the ISI for different neurons. So you should probably, it's not with your synchronization measure, you cannot really dissociate nicely effects in changes in synchronization from effects of changes in firing rate, probably, because you don't have a reference oscillation with respect to which to compute the phases. Well, it will work only if the firing of your neuron was tonic somehow. But,
right, all right.
So the kind of simulated pharmacology, the SSRI, and also the ketamine. So both of those were implemented only in the VACC module. Is that
Yes.
correct?
Yes.
So how do you think about that, right? Because so what's your kind of simulated deficit is this hyperexcitability of ventral ACC. A pharmacological agent presumably could be impacting both areas, and you need the persistent activity in DLPFC for your working memory activity or whatnot, right?
Yes.
And so if you're weakening persistent activity, yeah, that helps VACC being hyperactive, but also can hurt your DLPFC network. And people have talked about, so for example, one of the co-authors of the paper, Helen Mayberg, has done stuff on microstimulation, where they get kind of regional specificity in that way. So when you played with this, did you try a global perturbation, and did that not work? Do you need regional specificity?
So
How do you think about that?
First, for the SSRI, we followed what they did in the paper is that they changed the parameters in the VACC network. Yes, so that's why we were just trying to replicate this result. And when we tried to do for the NMDA, we tried to follow the same concept just to be able to compare the results.
Simulating SSRI only in
Yes, they did hyperpolarization for the resting potential of the excitatory neurons in the VACC network.
But do they have a scientific justification for that? Like why that the drug only acts in...
Yes, they cited some papers, like multiple papers, that say that the SSRI could be represented by this changing in the resting potential.
But only in a specific area.
Yes, me for myself, I was like wondering if this should change only in the resting for the VACC, and that's why if I had more time, actually I wanted to change the resting potential in the other network, and even to check the resting potential for the inhibitory neurons, and the same for the NMDA. But actually, because of the lack of time, we didn't do all the options. But yeah.
So also your synchrony values, okay, are very high in the baseline.
Yeah.
0.9. So do you think that if increasing the level of noise, or maybe reducing the drive, the results will be robust? Have you tried through the robustness of your results, reducing a bit the firing? Because it looks like it's nearly epileptic.
Yeah, we didn't try, but I think that if we decrease these inputs, it will be like less synchrony in the network, and also there's a lot of connections. Like all neurons are connected with all neurons. That there is now like they don't take into account like the distance. Like the neurons should be, in my opinion, the neurons should be like. If a neuron is far away from the other neuron, the connection shouldn't be like the same strength of a neuron that is really close to the other. So I think that there is a lot of things to implement in this work, and I think that because of all these simplifications, the synchrony is like really high in the network.
And what are the new things that you made with respect to the paper? So where the reproduction stops and your new things begin?
Yeah, so for comparison with the paper results, so actually we was not able to compare exactly with the paper because they stimulated for like 60 seconds, and for the 5 seconds it took a lot of time to stimulate just 5 seconds. But the general behavior we generated is like what was stated in the paper. However, for the question of the parameters, and I can see that there is like the fluctuations is for the synchronization, they are very high. So I think we need to play somewhat with the noise and with some of the parameters, because some of the parameters are not that clear in the paper. So just we wanted to like generate behavior that is similar to what they are stating, but yes, that noise and the level of noise, we need to play with more.
Okay, now we have expired the time, so let's go for lunch.
Okay, thank you.
Okay, the lunch will be shorter.